Very, very much, everyone who is here today. Welcome to the panel discussion on COVID-19, current pipeline and innovations for therapeutics and vaccines. This is BioCT's first foray into a virtual event. And again, we thank you very much for joining us for this important conversation. This webinar is being produced by BioCT in partnership with Advanced CT. Advanced CT is advancing business partnerships to engage, retain, and recruit companies in Connecticut. My name is Dawn Hocevar. I am President and CEO of BioCT, and BioCT is the industry voice for the bioscience industry. Before we get started, I'd like to go over a few housekeeping issues with Zoom. You should see an interface on your computer if you haven't done Zoom before. So hopefully you've taken the option to listen via your computer. Otherwise, you can be dialing in. The host has muted everybody. And please, audience, you want to stay muted as well as keep your video off. The panelists will come forth and center. Can everybody just, is my photo picture coming up large too, folks? Yes? Okay.
Looks fine here, yep.
Okay, thank you. So today's webinar will have a Q&A format. You will have the opportunity to submit questions during the presentation, and you can use at the bottom of your screen, you will see a chat button. So just click on that chat button and type a question in. And at the end of the panel discussion, Mostafa and I will look at the questions and try to get as many of them answered as possible. Today's event is being recorded and will be available for review as well. So at this point, I'd like to introduce David Campbell. David is the VP of Business Development at Advanced CT, and he'd just like to say a few words. David? David, you'll have to unmute yourself.
Sorry, let me try that again. I apologize. You would think after 40 dozen Zoom calls, I would have it down. Thanks again for Dawn. I'd like to make a few brief comments and welcome you on behalf of Advanced CT. We're thrilled to host this webinar in partnership with BioCT and Connecticut Innovations. Advanced CT is the statewide economic development organization. We work hand in glove with the Connecticut Department of Economic and Community Development and many other partners, including sectoral groups like BioCT, to recruit companies, startups, and entrepreneurs to the state. I would like to highlight several ways in which we are supporting the state's bioscience sector. First, bioscience is one of the priority sectors that we are focusing on in our recruitment efforts. Second, as part of the state's economic action plan, which is still in development, we've been analyzing bioscience and innovation trends in the state to form activities to strengthen the bioscience ecosystem and the overall Connecticut economy. Third and finally, we're working with BioCT on a nationwide survey being conducted by the Coalition of State Bioscience Organizations, CSBI for short. I believe this is the first time that Connecticut has had sufficient resources to participate in this important survey. Our Crackerjack research team has contacted the HR department at several biotech companies to ask them to complete the detailed compensation information that will be integrated into the national survey. And our super partnerships team has interviewed personally about a dozen biotech CEOs to understand their skills and human talent requirements and pipeline. This information likewise will be incorporated into the national report, as well as from each state that's providing the same information from their perspective. Because of COVID-19, I believe the survey is currently on hold. And we also were considering to attend the Bio International Convention this year to be held in San Diego. I believe that's been canceled. Next year, I believe it's again being back in Boston. Suffice to say, bioscience is an important part of our work and our cooperation with great groups like BioCT. Let me close by thanking each of the panelists for the great work they are doing on COVID-19. We appreciate it, and we certainly wish you every success. Back to you, Dawn. Thank you.
Thank you very much, David. And I will just say one brief thing. Yes, the in-person bio convention in San Diego has been canceled. However, Bio is still having virtual one-on-one partnering and virtual education information that you would normally see at the convention. So if you do want to get involved in that, please go to bio.org, or you can certainly ask BioCT for information and assistance. So thank you. Before I introduce the moderator, I just wanted to make note that Pfizer is not able to participate in the panel today due to rapidly emerging priorities in response to COVID-19 pandemic. They are simultaneously working on multiple efforts, and unfortunately, there are some legal sensitivities associated with public discussions at this time. They asked me to pass on their apology and express their appreciation for everyone listening today. Pfizer Connecticut leadership recognizes the critical importance of discussions like this, and they are hopeful to participate in an upcoming future panel. So with that, I would like to introduce our moderator, Mostafa Anna Liu. He is the Executive Director of Venture Development, Office of VP for Research and the Technology Incubator Program, TIP, at UConn. When he was at Pfizer, he was the site head for global clinical technology in Groton and New London, a division focusing on emerging technologies for development and validation of biomarkers and diagnostics for drug development. Prior to joining Pfizer, Dr. Anna Liu was Director of Oral and Maxillofacial Imaging Research, Associate Professor of Radiology at Indiana University, and Associate Professor of Biomedical Engineering and Electrical and Comp Engineering at Purdue University. He was also President and CEO of Thermometric Technology, Inc. He received his PhD from Purdue University, followed by postdoctoral fellowship at IBM, T.J. Watson Research Center in New York. Thank you all for being here, and I will now turn it over to Mostafa.
Thanks, Don. Good morning, everyone. I'd like to welcome you to our virtual panel today that is focused on therapeutics and diagnostics around COVID-19. This is not a surprise and well known that COVID-19 has posed a major global challenge impacting nearly everyone, everywhere. And in response to that, there are multitude of activities in number of sectors. And two particular areas that is greatly impacted. One is the public health as well as economy today. And some of these impacts, we see the short-term results of it, and there will be long-term association of these impacts that we will see coming up. In response to this, I think there are two groups that have been extremely active to address the challenges that we are facing. One is the healthcare providers that have been on the front end of this effort. And the other one is a group of innovators, researchers focused on therapeutics, diagnostics, and vaccine development that have ramped up their activities. I've been in touch with most of the panelists that we have today, and I can attest that in the past 3 weeks, they have been working on 24/7 to expedite what they have been working and get the outcome to the market, to the patient as possible. Today, we have 5 company institutions that choose to give you an overview of the landscape of what we have right now when it comes to diagnostics and therapeutics and vaccine, and what is in the pipeline, short-term pipeline, as we go through this. We have structured the sequence of conversation from diagnostics to therapeutics and vaccine, mainly because of the urgency of the need that exists in that area. And the flow of conversation will start with the introduction of each panelist, who they are, as well as what their organization and companies are doing. We will have set up a series of questions for them that I'll go through during Q&A part of it. And at the end, we open up the floor for audience questions. As Don mentioned, the only way to ask your question would be going through the chat on the bottom of your screen and type your questions there. we will not be able to get the questions either through video or audio. As I understand, there are excessively large number of people on the call today. With that in mind, I'd like to get started with Dr. Mark Adams of Jackson Lab. Mark, that's all right, Don, that's okay.
Hi, Mostafa.
If I could just say one more
glad.
thing, I forgot to mention to the audience that if you're seeing a lot of little boxes and you want to see us or the panelists up front, up in the top right-hand screen, please click on presenter mode and you will be able to see each panelist. Thank you.
Just to remind the panelists, this screen switching is sensitive to your audio. As soon as you start making noise, it's going to switch to your like. With that in mind, let's get started with Mark. Go ahead, please.
All right, thanks for including me on the panel. I'm Mark Adams. I'm the Deputy Director at Jackson Laboratory for Genomic Medicine in Farmington. And prior to the COVID-19 era, I directed the microbiome programs at Jackson Lab as part of the services group there. JAX has two broad areas of contribution to the COVID-19 response. One that I won't really be talking about today, but that some folks might be interested in is development of a human ACE2 mouse that's going through speed rederivation up in Bar Harbor, and orders are being accepted for those animals. The timeline is still some weeks away for availability, but I encourage anyone who might want to do in vivo studies with the mouse model to contact folks in Bar Harbor. There's information on the website. Here in Farmington, we have been working with the state on establishing SARS-CoV-2 testing for clinical specimens. That started about a month ago, and really through a remarkable series of dedicated efforts on the part of folks here at the State Public Health Lab, at the hospital partners that we've been working with. We've now been running this week about 200 samples a day and are working aggressively to try to increase that capacity. So Mostafa, I think I'll stop there in terms of introduction, and we can go through the rest of the panel and go to Q&A as you like.
Thank you. Thanks, Mark. Yeah, thank you, Mark. So as I mentioned, we are going to keep the sequence based on the diagnostic therapeutic vaccine. So Eric Kabrams is the next. Eric, go ahead, please.
Thanks, Mostafa. So just a little personal background. I've been leading technology development at 4Catalyzer in a few different areas in bioinformatics, brain-machine interfaces, next-generation imaging. And fast forward to about a month ago when the pandemic broke out, and our founder, Dr. Jonathan Rothberg, and the people at 4Catalyzer decided that we wanted to see what we could do, that what our group was uniquely situated to contribute in this fight against the pandemic. And what we identified was the need for a massive scale up of our testing capabilities. And to address that, we're developing right now an at-home test. So a full at-home test at which the user can collect their own specimen, perform the test themselves, and interpret the results themselves. And we're in parallel planning for success, scaling up the manufacturing and supply chain lines, and working as hard as we can to get this out there as quickly as possible.
Thank you. Next, we have Dr. Anil Diwan, President of NanoViricides. Anil?
Hello, Mostafa. So we have been working on developing a completely different approach towards antiviral diseases. What we have found, and I think everybody knows, that no matter how much viruses change genomically, they continue to bind to the same receptor and to the same site on cell surfaces. So what we decided to make is a polymeric micelle material that essentially looks like the cell surface to the virus. And we have that approach successfully implemented now for many different things. We have the shingles program is going towards an IND, should be filed within the next couple of months. And along the way came COVID-19 around December, I think it was the December 3rd week or January 1st week. We learned that it was ACE2 binding. And we discovered that in our molecular modeling, we already have viral ligands. So we started working on that. And I'll describe that a little bit more during the panels.
Thanks, Anil. Switching to Gary Mathias, the CEO of Oh boy. Gary, go ahead, please.
Thank you, Mostafa. Gary Mathias, I'm the CEO of Thetis Pharmaceuticals. We're based in Brantford, Connecticut. We're a biotech company developing resolvin-based therapies for acute and chronic inflammatory diseases. Prior to the COVID-19 pandemic, we have a candidate in preclinical development for inflammatory bowel disease with the support of Connecticut Innovations, the Crohn's and Colitis Foundation, and the National Institutes of Health. And that candidate is moving quite nicely. Resolvins are endogenous lipid mediators that help to orchestrate healing and resolution of inflammation. And as the pandemic, our knowledge of the pandemic emerged, and the importance of treating acute respiratory distress syndrome became very evident, we looked at the pharmacology of our lead candidate and realized that we had in our pipeline a drug candidate called TP317 that could make sense in addressing acute respiratory distress syndrome, otherwise called ARDS, which is the principal cause of mortality in COVID-19, characterized in particular by patients who develop pneumonia and then progress to ventilation, where there is a very significant rate of mortality. So we're undertaking a development program to get 317 into the clinic, we hope, by Q3 of this year. We have an extraordinary body of information related to its safety and its therapeutic potential that we think makes it a very interesting and unique entry into the treatment of what is a very important aspect of the disease in terms of what is causing people to die. Thank you.
Thanks, Gary. You know, we had suggestions to the audience what to do with the mute and other thing. As soon as I mentioned Gary's name, my phone started text popping up here. For the audience, please do not text or email your questions to me. There is a chat box on the bottom of the screen that you can send your questions as well as comments that you may have. As you know, this is a multitasking environment. We are dealing with a number of emerging issues within UConn Health as well as the companies that we have here. And my apologies for being distracted here. Now, moving up to Dr. Bijan Almassian, CEO of CaroGen. Bijan?
Good morning to Mostafa, for inviting me. I appreciate it very much. My name is Bijan Almassian. I'm the CEO of CaroGen and a co-founder of the CaroGen. CaroGen technology is based on the work of Professor Jack Rose at the School of Medicine. This is an artificial virus that he developed over the last 2 decades, along with the VSV system that has been used for immunotherapy and vaccine development. Out of this platform, which is we call AVIDIO, which stands for Artificial Virus for Infectious Disease and Immuno-Oncology, we're building a portfolio of immunotherapy teams in collaboration with Yale School of Medicine, as well as UConn, the School of Medicine, as well as Albany Medical College and Wayne State University. We have a portfolio of products in hepatitis B virus infection for developing immunotherapy for potential cure. We also working on a colon cancer with UConn Health. And over the last couple of months, we have begun working with Professor Jack Rose on COVID-19. Dr. Rose has a history of developing vaccines. He, in fact, developed the vaccines for SARS in 2003. He published 3 papers between 2003 and 2008. We're building what he has built, and right now he is leading the effort. All the work so far is being done in his laboratory, and we will give you an update when the next part of the discussions.
Thank you. Now we get into the questions section, and starting with Dr. Mark Adams of Jackson Lab. Mark, I have a 2-part question for you, and I'm sure that everybody has heard about all these PCR-based diagnostics that are coming up or being refined. would you please give the audience a landscape of what is available for COVID-19 diagnostics as of today?
Sure. So the qPCR-based methods are the first and still most widely available diagnostics that are available. These were initially, as I expect many folks know from either reading the New York Times this week, in which there was a cover story, or just listening to the news that testing has been an issue, identified early as a priority. The CDC developed a qPCR-based assay that was initially ineffective, and that led to a delay in the subsequently between the CDC and the FDA. There have been a series of relaxations of constraints on providing testing from a variety of providers. And so more and more folks are getting involved in this process, which is starting, but still just, I would say, starting to alleviate the need. The demand still far outstrips capacity for testing. That's for a number of reasons. First, of course, these have to be performed in CLIA labs. Just give an example of our CLIA lab was initially licensed in 2013, but has been doing all cancer diagnostics and genetics. It's a separate licensing process for virology. And so we work very closely with the state health lab in order to get that certification to be able to perform COVID-19 testing. Initially, there were only a few vendors for supplies and kits for qPCR. There are now many. I looked last night, I believe there are over 230 emergency use authorization requests that have been submitted to the FDA for testing kits, and at least 20 of those have been granted. And in addition, well over 100 labs are using laboratory developed tests that go through a different process for validation and confirmation in order to be performed. Some of the large testing companies, Quest and LabCorp, are using high capacity instruments, machines from Roche to do the assay. But those large testing labs have long in terms of clinical practice, turnaround times. 5 to 10 days is typical for getting a test result back. So, in addition, there have been reports in different areas of issues with getting access to either laboratory reagents and supplies or increasingly testing kits. Many of those restrictions or alternatives are being developed in many of those areas, but I would say that it's still quite a rocky landscape around getting this testing done, and many groups in many areas, whether that's all the way from the large commercial testing companies down to academic labs that have the ability to implement these processes in a newly certified CLIA environment, are working very, very hard to increase the testing capacity. Just one more point on that, and to echo Eric's comment earlier, there may be a peak of infections coming, but there will not be any peak of testing coming. I think that certainly as we make attempts to come back to normal in terms of the economy, in terms of healthcare, in terms of a wide variety of personal interactions, there's going to be a large and continually increasing demand for testing to identify people who may have been exposed, who have symptoms, and get them appropriate treatment and isolation. And so I think this is an aspect of the coronavirus world that we're going to be living with as an evolving landscape in this for quite a long time to come.
Thanks, Mark. I think one key phrase to remember what Mark mentioned about EUA, emergency use authorization. This is a keyword that we are hearing so frequently. And in addition to the PCR-based technology, qPCR, RT-PCR, there are other innovative methods are being developed. You perhaps saw the press release coming from UConn Health. That's a CRISPR-based approach. Still needs to be validated, but the landscape is quite promising. And additionally, to me, the surprise was there is a clear indication that CT, computed tomography, can detect some of the lesions for the pre-symptomatic patients. And I looked at some of these CT images myself yesterday, and it clearly shows a ground glass opacification, which is asymmetric toward the peripheries. And that by itself could be another add-on tool if there is a lot of surge for demand for other type of diagnostics. Mark, I want to switch to the second part. You already mentioned some of the things that JAX is involved. JAX is known for its mouse work, and with the footprint that you have in Farmington, Connecticut, you have expanded significantly in human health. Can you elaborate further about technical capability, capacity, and thinking within JAX lab around COVID-19?
So JAX is approaching this from really a pretty broad range of angles. So the mouse group up in Bar Harbor, as I mentioned, is breeding up humanized ACE2 mice, and those will be available for a variety of preclinical testing sorts of experiments. As folks may know, propagation of the virus, either in tissue culture or in animals, requires a biosafety level 3 facilities, which are not very widely available. Certainly not, we don't have them at JAX anywhere. I'm not sure there's anything at UConn. They're very high overhead kinds of facilities. So there are a lot of restrictions in how that's the kinds of studies that can be done based on the limitation of those facilities. So both here and I think many other places, we're working on a variety of ways to study the virus without propagating it, whether that's in by breaking apart the virus into its individual components, looking at the specific signaling pathways within cells, and so on. And I think there's a lot of research that can be done without that kind of BSL-3 environment.
Thank you. Now switching to...
I had a question that came through privately about the definition of CLIA. So CLIA, a CLIA lab is the, oh gosh, I had it and then I'm blanking on it.
Clinical lab something authorization.
Authorization, yes. It's the nationwide certification process for performing tests where results will be returned to patients. And it covers a wide variety of systems for defining standard operating procedures, standards for how tests are conducted, proficiency testing, accuracy, validation, verification procedures to make sure that results that are returned to patients are useful and valid and informative, reproducible.
All right, so clinical lab improvement amendment, CLIA, and that's against CLIA lab.
You know why people use
CLIA lab terminology. Along with EUA, you will hear that very frequently as this novel test pops up and requires a specialized, localized facility to do that. So we are going to hear that very frequently. I'd like to switch to Eric. Eric is from 4Catalyzer. He mentioned already Jonathan Rothberg, that you remember as a pioneer in next generation sequencing, and he man behind portable ultrasound as well as desktop MRI. But Eric has taken another initiative, hoping that we can do at home kind of diagnostics. Eric, please go ahead.
Sure. So the way I like to describe our project is Dr. Jonathan Rothberg and our team has been developing machines for DNA and RNA sequencing for over 20 years. And this time when the pandemic broke out, Jonathan said to everyone, let's do it again, but this time without the machine. So what we're trying to do is we're developing a test that can be run completely in the home setting by untrained users with no special equipment. And just to put the test in perspective for the audience, when we look at the testing landscape, we like to distinguish tests along two characteristics. So the first is the underlying science behind the test. So that is it a molecular test that's detecting the nucleic acid of the virus itself? Or is it an immunological antibody test that is testing not the virus itself, but your immune system's response to the virus? And so our test is a molecular nucleic acid test. And then the second feature that is, I think, most important for placing our effort in the spectrum of all these complementary efforts is the process of running the test. So who collects the sample, who runs the test, who interprets the results, who decides what to do about it. And there really is a spectrum. And so on one end of that spectrum, you have the traditional testing laboratories where a trained technician collects the sample, runs the test, and interprets the result. Then coming into the middle, you have self-sample collections tests that are just starting to develop right now, where the user collects the sample on themselves and then hands off the sample to a trained technician, or perhaps mails it to a testing laboratory for them to perform the test and interpret the result. And in this area, I'd like to highlight some amazing work that's been done by the Gates Foundation with the University of Washington and some other partners that's just coming out that develops that that demonstrates that self-sample collection with a simple nasal swab rather than the gold standard nasopharyngeal swab, which is quite difficult for users to obtain. But this work shows that the simple nasal swab is actually of comparable efficacy to the previously described gold standard. And then on the other end of the extreme, there's a home test, which is what we're developing, where the user collects the sample on themselves, runs the test for themselves, and then interprets the results. And so in our development work, there's two stages. And so there's developing the assay itself. And then there is, how do you map this into a form that is easy for anyone to use in their own home and manufacturable at a massive scale? So we're doing everything in parallel and iterating very quickly, but the test that we're developing right now is an at-home molecular test.
So Eric, many people think this is a wishful thinking, that a small company in that corner of Connecticut can develop such a test at home. Perhaps you need to elaborate what you guys have done with those really, really unbelievable project on portable ultrasound and desktop MRI to give some credibility that you guys can do it. You have done it and you can do it again.
Thanks, Mostafa. So first, I think we absolutely can do it, and we are doing it. Just to give some background on the older companies in this family. So Mostafa is referring first to Butterfly Networks, which that company has done an incredible job of creating the world's first whole body ultrasound on a chip device. So it's a single handheld ultrasound scanner that can do the whole body, and it doesn't require an additional cart, and it can be operated from a smartphone. And it uses machine learning to make it easy for untrained people to acquire images. And this ultrasound scanner has been released now for over a year and is now the best-selling handheld ultrasound scanner in the world. And I also want to highlight Butterfly's efforts in the pandemic response. So along with lung CTs being shown to be very useful in diagnosing and managing patients with COVID, in addition, there's evidence emerging that lung ultrasound is also a useful way to monitor COVID patients. And so that's Butterfly Network. And I also want to highlight another company, Hyperfine, which just 2 months ago received FDA approval for the world's first point of care MRI device. So this is a whole MRI system that is on a cart that can just be wheeled into the room and plugged into the wall and operated off of an iPad. And again, this point of care MRI system is playing an instrumental role in some radiation departments now, where it's very important that we keep safe distancing and take special measures to avoid contamination. And so having an MRI system that you can bring to the patient is really useful. So those are just two of our older sibling companies that I hope should lend some credibility to our efforts.
Thanks, Eric. Back to your at-home approach, are you envisioning that being a quantitative method or yes or no type?
First, yes or no. I think it could be the case that it could be used for quantitative, but definitely what we will develop first is a binary yes or no.
Thanks. Now, moving on to Gary, CEO of Thetis. Gary, your company was focused on IBD, and that is one of your leading products that you have there around resolving. Can you, especially when you look at the impact of COVID-19 on lung, short-term and long-term, can you elaborate, how did you get to COVID-19 and what evidence do you have that your resolving approach can be helpful in this area?
Sure. So I think it's important to just put in perspective the importance of treating ARDS, acute respiratory distress syndrome, and just some facts that have come out of China. The one study, an initial study, showed that approximately 42% of COVID-19 hospitalized patients develop ARDS, and of these, 50% die. While the press is talking about the fact that we will have a surge in hospitalizations and in deaths in the near term, it is expected that there will be a rebound in COVID-19 in the fall and winter before a vaccine is likely to be developed. And therefore, the urgency around treating acute respiratory distress syndrome will continue. The underlying research supporting the use of resolvin therapy in ARDS comes from work at the Levy and Sirhan laboratories out of the Brigham. And this is work that Charlie Sirhan is a director of a lab that does therapeutic work at the Brigham, and he discovered resolvin E1 almost 20 years ago. And then Bruce Levy, who is a pulmonologist, carried out work to demonstrate that resolvin E1 has pro-resolution and tissue reparative effects in a number of mouse models of ARDS. So the underlying research is there. And as we looked at the action of 317, which delivers resolvin E1, we recognized that there was an opportunity to bring forward a non-immunosuppressive approach to a disease that has no approved therapy. And I want to underline the following. ARDS is a disease or a condition that afflicts almost 250,000 people a year in the US, independent of COVID-19. It is a critical unmet medical need. There are no approved therapies in ARDS. So what we see here is both a need in the context of COVID-19, which is expected to continue, as well as an overarching need to address the problem of ARDS in the general population.
Thank you very much. This is one of those exciting topics that Gary is working on, and a number of groups are looking into that to figure out a way to put it on the fast track and help the company to move it forward as fast as possible. Anil, in NanoViricide, you already alluded to your platform. I have the same question. Can you elaborate more about your approach and the evidence that you have? You think it's going to be effective in this area, and share with us the timeline and the development plan.
Sure. So that's a lot of questions. I don't think you have muted yourself. I'm not showing up on the screen. Am I showing up on your screen now?
Yes, yes,
Oh,
you're fine, Anil.
So that's a lot of questions. Let me start with the beginning. So around 2014, when the MERS coronavirus epidemic broke out, at that time, we were actually asked by Public Health England to look into it and try to develop something for that. And so we had done a lot of molecular modeling around coronaviruses at that time. SARS coronavirus, as you know, was from 2002. So there were structures available. When this, like I said, towards the end of December or January 1st week, when this thing came out, that ACE2 is the virus protein to which SARS-CoV-2 is binding, I looked at my previous molecular model structures and I dumped some of them in there. And we were very, very lucky that we found a few structures that actually did bind exactly in the space where the S protein, the spike protein of the virus, binds to the ACE2 receptor. And that's very important for us, because that means that we will be not only attacking the virus particle, but also ensuring that when the virus changes genomically over mutations or other exchanges, it will still continue. It still continues to bind to the same receptor the same way. So it will, our drug will still continue to be effective. And that's very, very important virology, because viruses mutate away and escape drugs very readily. Influenza, of course, is a hypervariable virus, and so is HIV. Those cases are well known, but same thing happens with dengue viruses and other viruses as well. So we developed these things. We were lucky that many of them were already synthesized in parts and they were on our lab shelves. So we went ahead, and our whole approach is that we take these virus binding ligands that mimic the receptor, and then we attach them to a surface, polymeric micelle-based surface, so that it looks like a cell membrane to the virus with high density of receptors expressed on it. So the virus would specifically bind to our drug rather than to cellular receptors. When it does, the material that we are using is not a nanoparticle in the sense of a hard particle. It's a soft material. So it has lipid inside, and it has a polyethylene glycol coating corona made up of the micelle. And so what happens is when the virus does bind to it in multiple spots, then the lipid-lipid interaction between the surface of the virus and the inside lipid of our material takes place. And this micelle that we have literally coats over the virus, encapsulates it. And in electron micrography, we have seen that that destabilizes at least some virus kinds of virus particles completely. So that's the general approach. We have applied it to many, many different viruses. Our lead candidate currently is in herpes viruses family, the shingles virus, VZV. It's ready for an IND. And the ligand that we found for this particular virus, the coronavirus 2, is closely related to that ligand. So we are very far ahead in terms of the CMC aspects and CGMP manufacturing aspects. I believe people here know that we built a facility in Shelton that can do CGMP capable manufacturing in multi-kilogram scale. So that would be sufficient to supply as many as 1,000 patients per batch or so. And so we are already there in terms of that perspective. We have started testing our candidates in-house. We have a BSL-2 virology lab, Connecticut state certified. And there we have brought in the available viruses, the circulating coronaviruses that can be used in a BSL-2 lab. One of those viruses actually binds to ACE2 receptor, uses the same receptor. So that serves as an appropriate surrogate model. And then another virus binds to a similar receptor, APN, which is also peptidase, and it's in the same superfamily, it looks like. So we are assessing whether our drugs are sufficiently broad spectrum that they would attack other coronaviruses in addition to the existing one. We are now working on a collaboration, setting up a collaboration to be able to test against the COVID-19 virus itself, the SARS-CoV-2, which requires BSL-3 facility or BSL-4 facility. So that should be, the MTA, material transfer agreement, should be completed within the next week, and we should be advancing the drug to test against that. In the meanwhile, we have also commissioned an animal study. As you know, Jack's Labs already stated there is no appropriate animal model for this particular virus. That's true. But the NL63, the coronavirus that is circulating, that also binds to ACE2. And similar to the work done by another group in when they were developing remdesivir and testing it in animals again for activity against coronaviruses, we were able to develop an animal model by using extremely high infection of the virus directly into lungs, of the virus particles directly into lungs. That does mimic some portions of the disease, but the infection is so high that probably the mice are never going to be able to recover, which was the finding in the previous study that I referred to, also Dr. Barrick's group study. So our drugs are being tested there right now. As I see, we have received initial data. Now, of course, we'll have conferences with the people who did the study, principal investigator, and understand more about it and put that next study in place. So we are doing everything. This is a completely novel drug against this, unlike what other things are going on, which are drugs that were already existing and had some safety toxicology already developed, and they are going into clinical trials now, which did not have, from FDA's perspective, FDA essentially waived the requirement that the drug should be effective against coronavirus in some model for some of the clinical trials that I have seen. So we are actually looking for a drug that is effective against coronaviruses. And SARS-CoV-2 is today's, yes, SARS-CoV-2 is probably going to continue even after peaking into the fall. That is the current estimation. It's going to become a circulating strain. SARS-CoV-1 did not because it just got killed off because of its high lethality rate. It did not have a way of circulating. However, other coronaviruses have become circulating strains. They cause common colds in us, in humans, and they do not cause the severe pathology. Some patients may have ARDS developing from even those, but most patients do not. and we recover. So there is the potential for this kind of drugs going year round, and we are now focusing our attention to develop these drugs. Right now, we are doing exploratory, highly parallelized, but a quick type of approach, which we should be able to get to if we get funding. We should be able to get into humans in about 6 to 8 weeks. That's our estimate, because that's basically the timeline for completing one initial set of safety toxicology study. And manufacturing we are doing in parallel. So, however, we do not have that kind of funding right now. And we have to obviously bring in outside collaborators for everything, and that costs money. So we are waiting for funding. We will start working on that. We have started working on that. As you know, Senator Blumenthal visited us, and he has said that he is going to, not only us, of course, but Connecticut companies, he is going to help get the BARDA funding that was recently gone into law.
Thanks, Anil. Again, for those of you who may not be familiar with BARDA, B-A-R-D-A funding, that's a special mechanism that they operated based on DC around public safety and pandemics that it was around. And they have ramped up their activities, and there are a number of funding mechanism, collaboration mechanism are coming up from that direction. Now, moving on to Bijan. Bijan, you have been developing this novel virus-like vesicles, VLVs, especially around, Anil, can you mute yourself, please?
Hello?
Can you mute yourself?
I'm sorry,
That's all right. And Bijan, you need to unmute yourself so we can see you full screen. So go ahead about VLV, hepatitis B that you're working and how it's related to COVID. Bijan, we cannot hear you.
Basically, our approach is very different than many other approaches. Here we are fighting a virus with another artificial virus. And this VLV, or AVIDIO, which stands for Artificial Virus for Infectious Disease and Immuno-Oncology, was discovered at Yale by Professor Jack Rose, who is also a co-founder of the company and the chairman of the Scientific Advisory Board. Dr. Rose developed a vaccine for SARS in 2003 or 2004, using his own VSV platform that he discovered at Yale. As you may note, Mostafa, VSV also was the platform that Merck has used to develop Ebola virus, the vaccine, which was approved in 2019 in Europe as well as US. So we have been working on this artificial virus for over 5-6 years. Our initial work was on developing an immunotherapy for treatment and potential functional cure of patients chronically infected with hepatitis B virus. And then we have expanded to the oncology. This virus is also, this artificial virus is oncolytic, has the ability to deliver multiple antigens. Because it's artificially made, the component of this virus comes from different viruses that they are not commonly infect humans, so there is no pre-existing immunity for the patients or volunteers who received it. There are a number of advantages of this VLV over the existing platform. One, its ability to deliver multiple antigens. So there are a number of antigens in COVID-19. Of course, the major one is S. There are also another, the antigen is called N, or nucleoprotein. And the other advantage of this platform is self-adjuvanting. There's no need for adding adjuvant to it. So the platform has broad application in infectious diseases. It could be used as prophylactic because you induce antibody against the antigens. And also you can basically induce T cells against the antigens of interest. We have validated this platform in hepatitis B virus, different animal models, acute models, as well as chronic models. So it could be used for prophylactic as a vaccine. It could be used for therapy. Here for COVID, why we're interested in COVID-19 vaccine, because again, it's based on the knowledge and experience that Dr. Rose providing us. He has begun developing a vaccine for COVID-19. I'm very pleased to report to you in a very short period of time, we have already built the vaccine. We have tested in terms of the quality control of this vaccine to make sure it does what it's supposed to do, expressing the S antigen. Of course, we use different type of S sequence. We believe there are different sequences that might give a better immunogenicity. And right now, we have characterized the expressed S antigen to make sure that it's appropriately glycosylated, which that needs to be. And then at this time, we're doing the immunogenicity. We're in the middle of that immunogenicity testing, and we believe within the next 4 weeks, we will have sufficient data to advance our vaccine to the next stage of development. And also, we have begun our discussions with FDA for the hepatitis B virus. We had a very good pre-IND meeting with FDA. We understand what the requirements are for the manufacturing, preclinical, and we're developing a clinical protocol to be used for this COVID-19. Like the other companies, we're a small company. We are badly in need of financial support to make sure that we stay within the timeline. It's all depending on, we have established relationship with contract manufacturers who have experience with similar products. We believe we could skip some of the preclinical studies that usually are required by FDA and get into the IND as well as initiation of the clinical trial.
Thanks, Bijan. You can go on mute now, please. I noticed that Dr. Patrice Milas is on the call, and I'm going to pick on her. Patrice, she is one of the pioneers in next generation sequencing in drug development and diagnostics. Patrice, if you can hear me and if you can unmute yourself, I think you have been following this sector very closely. I want to get your take on the diagnostics. Where do you see the most emerging solutions related to COVID-19? Go ahead, please.
So, hi everybody, Patrice Milas here. I'm in Rhode Island, so I'm not a Connecticut person, but I used to be at Pfizer. So we've been watching very closely the emerging diagnostics. And I think Mark, I forget who gave the great view of the laboratory developed tests, the UA approved tests and so forth. Overall, the ability to rapidly test an individual for COVID-19, it has emerged as something very key for our states, and the challenges associated with that are well known because, you know, the CDC test that was first put out required qPCR of 3 regions of the viral genome. One of those regions didn't work, so they went back and said, okay, only use 2 sets of probes. It's a very long test. It takes, you know, several hours to run, all the way to now, which are relatively rapid. The Cepheid being a large instrument platform that some of the hospitals have, that they've now transitioned to using the Cepheid cartridge, a 15-minute test. And then the Abbott is the test that CBS is beginning to bring online in these mobile testing sites. So CBS opened up a testing site at Twin Rivers Casino parking lot here in Rhode Island. I think it was Monday. They're doing the same in Lowell, Massachusetts, and they're doing it in Georgia. So that's about 1,000-person testing capacity a day at those sites, but it still doesn't approach the level. So Rhode Island is doing about 2,000 tests a day now throughout the state using a variety of different platforms. and a variety of different times, but it's really key. But that just addresses whether an individual has the virus at the time of infection versus the serology that everybody is interested in to understand all these asymptomatic individuals, which there are many, it appears, that we could then identify those as having had the viral infection and looking for IgG and IgM antibodies to say these people have been exposed, they've elicited an immune response, and potentially offer a way to bring people back to work, introduce them back into their workplaces. I've suggested it would be fascinating to think about workplaces being able to actually do the testing on site. So imagine if Brown University had the ability to test both for the virus and for antibodies to the virus. Could that be a way to keep students and faculty safe on the campus? So a lot of very interesting questions here. And it all relates to how good are the tests? Are they sensitive enough? Are they specific enough? False positive, false negatives. And we're learning every day as we go along and following the literature. So that's what I would just add at a high level, Mostafa.
Thank you very much. Patrice, you can go on mute, please.
Okay.
So Dr. Milas was not part of the initial panels that we put together, but we were quite lucky to find her online. For those of you who are developing diagnostics, she is one of the best resources you can find around us, and she's kind enough helping a number of innovative companies. With that in mind, I think we wrapped up some of the prepared questions here, and I want to switch to audience question, and I'd like to invite Dawn back to start the questions, please.
Thank you, Mostafa, and thank you, everybody. That was just a wealth of information. I'm going to start with a question to Gary, that you have an interesting compound, and they'd like to know, can you comment on the route of administration? Is it, IV versus inhaled, and the use in ARF prior to ARDS early prophylaxis?
Great question, and thank you, Dawn. We're currently working on developing 317 for intravenous administration. There is strong data suggesting that IV injection can lead to sustained elevated levels of resolving E1 in the lung. We also believe that an inhalable form may make sense down the line for more of a prophylactic use with life cycle management. But for purposes of our initial entree into the clinic, we do believe IV would make sense. In terms of the clinical trial that we would undertake early on in a phase 1/2 format, we're targeting COVID-19 patients with confirmed pneumonia by X-ray or CT who are not yet intubated. We believe there are real challenges in terms of getting informed consent from patients that are intubated, and that can be obviously a challenge for enrollment purposes. But we also believe that from a clinical benefit standpoint, that reducing progression to ARDS is critical. But we also see down the line an application for treatment where patients who have mild to moderate ARDS can be treated to prevent progression to severe ARDS when mortality rates rise precipitously. So hopefully that answers the question.
Thank you. Next question is for Anil. Is delivering ACE2 receptor decoy as nasal or inhaler can be used as prophylactic for healthcare workers on the front line to treat COVID patients?
It possibly could be, and we have not looked into that possibility yet. Prophylactic is a very difficult pathway for drug development, as you know. Something that is after infection and for treatment is actually a faster pathway for drug development, and we see that as the fastest route to get it to patients.
Thank you. Okay, I see a message for CaroGen. ADE issues have been seen in some previous SARS and MERS vaccines. Does the VLV approach negate these concerns? And what about other approaches using VLPs?
We are currently addressing that because really there are not the system, the preclinical system have not been established for COVID-19. The different investigators use different animal models. In fact, Moderna went to clinic without doing any preclinical studies, but Moderna had the previous drugs in the development, in different phases of the development, so regulatory authorities were definitely comfortable with the platform. We believe we need to do additional studies to make sure that we don't run into that difficulty. So far, we have not seen from all the different indications that we have been working on and the different animal models we have used, acute and chronic and transgenic mice, we have not run into that difficulties. So that's my answer.
Thanks, Bijan. There was a quick question that I can answer that before we go to the next one. It was about the use of the CT that would be limited. That's very true. You know, we have almost every major hospitals have multiple CTs, and we are talking about chest CT, not a full body CT. The challenge here is the radiation. And with all these PCR-based and swab-based methods, you can test the patient as many as time you want, but you know, there is a limit how much you can put people, especially asymptomatic people, into the X-ray machine. Now, I'm going to read a question that came through text to me, and I know who sent it to me. I told you not to text me, but since you asked that question, I'm going to pose it. Question is for you, Mark, that you mentioned that the diagnostic issue around COVID is not a today's problem, it's a long-term challenge, it's a long-term project. Can you elaborate on that?
Sure. So today, a patient either is tested through a drive-through center or in the hospital for acute, where an immediate answer is necessary. There are really very few solutions. The Abbott machine, the Cepheid machine that are just coming online now and are not widely available, those point-of-care devices are still very limited in their availability. I see that changing over time. But what we're doing at JAX is we've partnered with more than a dozen hospitals in the area here to provide pretty fast turnaround time for samples that come from the hospitals, mostly with inpatients. So we are receiving samples throughout the day from these hospitals. We're running 2 shifts, and so far we have declined to accept more samples than can be processed in a day. So unlike the big commercial labs that say, yeah, bring them, send them on, we'll bill you and we'll get the data to you when we can, we're trying to preserve that fast turnaround time. So all of our results have been delivered within, I believe, 16 hours of when we receive the samples. So that really helps the physicians who are trying to manage the patients make those critical choices about who needs isolation and what approaches are necessary for those. So obviously point of care will help in making those real-time choices. As we start to think about, you know, who else should be allowed into a clinical environment for testing, I'm not, you know, I think there's a whole variety of different scenarios that are going to be needed in terms of rapid tests or also routine tests that that might be used for monitoring people who are in a work environment. And I think the serology is also an interesting area to think about and look at. The models that I've seen, and if the country is beginning to get a handle on flattening the curve and limiting the number of transmissions this summer, really, you know, 95% of Americans are not going to have contracted coronavirus this spring. And so the serology alone, I think, is not going to be a solution for bringing people back to work and certifying a large portion of the population as safe, assuming that the presence of of antibodies means that someone can't be infected again. So I think there's a multifactorial approach that's going to be needed that will involve some combination of large testing centers, specialized testing centers, point of care devices, and if at-home testing comes online, there'll be a role for that as well. There was a comment about how all of this information is going to be integrated, and I think that's a really excellent question too. If right now all of the tests that we're doing, and certainly through point of care, the state health lab is collecting information on the positives, who they are and where they are. Really, as I understand it, the capacity to do contact tracing, though, is really still quite limited given the overwhelming demand based on the number of people who test positive. So the infrastructure around how test results are used is, I think, also a real opportunity for the near term to get planning on how that information is used to understand the spread of the virus, but also identify potential people who are at risk.
Thank you, Mark. There's a question here, is Thetis or anyone else focused on developing combinations using drug repositioning approaches?
I can speak for Thetis and say the following. We recognize that standard of care is evolving quite rapidly in the COVID-19 circumstances, and therefore we need to be prepared for clinical trials that will test 317 over other therapies that are being tested in clinical trials, as well as agents that are being repurposed. And I think anyone who is looking at immunomodulatory therapy to address ARDS or other sequelae of COVID-19 needs to be prepared to think about testing their product in that context.
Thanks, Gary. Eric, a question came for you regarding your home-based approach, meaning that have you explored the mechanism by which end user can report their results of home testing?
Yeah, that's a really important question that we're thinking a lot about. So I wanted to add something that I didn't emphasize in my initial description, is that our home test is accompanied by a companion mobile application. And so this mobile application is used to instruct the user on how to perform the test, and then also collects basic demographic and symptom information from the user, as well as the test result. And when the user is done with the test, all of this goes back to our cloud-based software infrastructure. And we collect all the results and package it in such a way to facilitate fast and scalable reporting to the relevant federal and state health agencies. So that's those are our current plans. And we are in discussions with the CDC and FDA on the optimal set of information and figuring out what would be most useful and in what format. But this is something that we're thinking a lot about.
Thank you very much, Eric. I see a question here for CaroGen. It seems like part of this might be a repeat, but what is the scale-up time for human ACE2 transgenic mice? Do these mice reproduce in 3 months? And how many pups in a litter? Are these mice the limitation in developing a vaccine? Is there anybody who would like to answer that? I'm not sure who it was for.
The first part of that, I can answer a little bit about the scale up of producing the transgenic mice. There are a variety of tricks and techniques that the labs up in Bar Harbor are using to expand the number of founder populations through embryo transfer and in vitro fertilization and so on, to expand the colony as fast as possible. So as I said, those are still some weeks away from being available, but at that time there should be quite a few. And I would leave it to someone else to describe how they would be used in vaccine development.
Let me address the one question before that was raised by one of the audience was the difference between a VLV and VLP. VLPs are proteins. The VLVs are packaged RNAs. VLVs induce broader spectrum of immune response, including antibodies, T cells, and innate response. VLPs in general, they induce antibody. In regard to the transgenic mice, I understand, in fact, this morning I spoke with my colleagues at Yale. Their discussions with Jackson Laboratories when that transgenic model is being available. But right now, the focus is on getting the immunogenicity studies and studies, and hopefully have some line of communication with the FDA to see whether that information is sufficient to start doing the GMP manufacturing and file an IND while we're looking at the preclinical model, examining in different preclinical models. FDA allows these things to be done in parallel, and we're planning to do the development based on this rush to the clinic as basically vaccines are urgently needed. As I understand, there are 41 different type of companies and investigators are developing vaccines for COVID-19s, and hopefully we will be one of the one to get to the clinic and providing some data. On the other hand, also we're validating a platform that is novel, is patented, and has not been in the clinic before for potential use of future pandemic diseases.
Thanks, Bijan. I see a number of questions popping up, and interestingly, some of the experts online are answering those questions, which is really helpful. I noticed that some of those questions are very specific ones, and it might be better to take it offline for one-on-one, that Don's office can connect you with the right players here. Before we conclude, I want to mention that this is one of many events that BioCT and others are putting together in state. Another topic that we did not discuss here is the issue of funding and financing of these technologies. And we are considering doing another event that purely focused on investment side of innovation. With that in mind, I'd like to thank BioCT and AdvancedCT for organizing this and all the panelists for joining us. And I'll pass the baton to Don.
Thank you very much, Mostafa. I'd like to thank you, the panelists, and everyone for attending today's webinar. The panel shared great insights on the work that's being done here in Connecticut to fight this pandemic, and we greatly appreciate the time that you took today to be with us. BioCT has a wealth of information available on our website. We have a landing page, and it is real time. We are constantly updating it, etc. So please go there if you're looking for information. If you can't find it anywhere, let us know and we'll try to help you track it down. We do plan, as Mostafa just mentioned, to do more virtual events. Hopefully you all found this valuable. And with that, make sure that you're getting our news alerts, because that's how we announce a lot of our events. And if you aren't on our news alerts, which come out, you can go to our website and sign up for that. Once you leave today's webinar, you will receive an email to take a short survey to please let us know any insight about this webinar, what worked, what didn't, and any recommendations you have for future topics. And you will also receive a link to today's recording. So on behalf of BioCT, AdvancedCT, our moderator, and our panelists, you all did a phenomenal job. Thank you, everybody, for being part of this today. And as our governor says, and we agree, stay safe, stay home, and stay well. Thank you all very much.
Thank you, Don. Thank you, panelists.